Okay, let's dive right into this. Welcome to today's explainer. We are embarking on a really deep, systemic exploration of the microbiota gut-brain axis. And look, we aren't just treating the gut and the brain as these separate, isolated entities anymore. We're mapping the continuously interacting biological network that literally governs our metabolism, our energy regulation, inflammation, recovery, and even our cognitive performance. We have to start treating the human body as one infinitely interconnected system.
So it brings up a pretty provocative question, right? Who is really in control of your mind? I mean, we typically view cognitive function as this purely top-down neurological process. Brain tells body what to do. But to truly understand human physiology, we've got to map the bottom-up pathways too. How in the world do 100 trillion gut microbes actually dictate our neurobiology, our systemic stress responses, and metabolic adaptation? We are looking at a living biological network where microbial ecology directly shapes mammalian behavior. It's fascinating.
Here's the agenda for today's explainer. First, we'll cover the microbiota gut-brain axis, then we'll look at breaching the intestinal barrier, followed by short-chain fatty acid signaling. After that, we're getting into the pathophysiology of irritable bowel, then neuroinflammation and mental health, and finally, diet, psychobiotics, and interventions. All right, kicking things off with Section 1, the microbiota gut-brain axis. Let's start with this concept of eubiosis. Think of this as our biological baseline. It's this state of perfect evolutionary harmony between host and microbe. In eubiosis, our microbiota is doing its job, supporting nutrient absorption, immune regulation, and metabolic homeostasis. But contrast this against dysbiosis, which is basically a state of microbial collapse. When this delicate ecology gets disrupted, it doesn't just cause a little localized digestive discomfort, It triggers a massive cascade of systemic dysfunction, altering literally everything from insulin sensitivity to neurotransmitter synthesis. Now, the gut and the brain are communicating continuously, and they do this through three main highways. First up, the vagus nerve. This provides direct, super-fast, bidirectional neural circuitry. Second, you've got the endocrine system. This relies on hormones like GLP-1 and PYY to regulate appetite, satiety, and energy balance, which are absolutely crucial mechanisms for any GLP-1 companion lifestyle. And third, the immune system, which uses cytokines and pattern recognition receptors like TLR4 to transmit inflammatory data. These domains are constantly exchanging real-time data, creating these incredibly complex interlocking feedback loops. Moving on to Section 2, breaching the intestinal barrier. Let's map out the exact chronological cascade of how psychological stress physically degrades your gut wall, because it actually starts in the brain. Psychological stress activates the HPA axis. The hypothalamus secretes corticotropin-releasing factor, or CRF. This then signals the adrenals to release the stress hormone cortisol. And here's the kicker. High chronic levels of systemic cortisol physically degrade the tight junctions of your intestinal epithelium. Once that barrier breaks down, bacterial endotoxins, like lipopolysaccharides, LPS for short, translocate directly into your bloodstream. So we've just traced a direct causal chain from emotional stress all the way to physical toxemia. Comparing these states molecularly is pretty wild. An intact barrier features a really high expression of tight junction proteins. Think claudins and occludins. It acts like a fortified wall. But in a compromised, leaky state, that LPS breaches the barrier and enters your systemic circulation. It then binds to toll-like receptor 4 on your immune cells, and boom, it triggers massive, systemic, low-grade inflammation. This inflammation impairs metabolic adaptation, messes with body composition, and kicks off a nasty negative feedback loop that just further damages the gut lining.
Okay, Section 3, short-chain fatty acid signaling.
Let's talk about the primary short-chain fatty acids, acetate, propionate, and butyrate. These are produced when our microbes ferment dietary fiber. Notice their actual mechanisms here. They bind to free fatty acid receptors like FFAR2 and FFAR3, which directly drives appetite control, insulin sensitivity, and GLP-1 secretion. But butyrate goes even further. It acts as an HNDC inhibitor. What that means is it provides epigenetic modulation, literally altering how our DNA is expressed, while simultaneously differentiating regulatory T cells to suppress inflammation. I mean, this is biochemistry at its absolutely most profound. 500 to 600 millimoles. Try to wrap your head around that. That is the sheer volume of these short-chain fatty acids your gut produces every single day through the anaerobic fermentation of dietary fiber. At this massive scale, your gut microbiome isn't just some passive digestive chamber, it's operating as a massive endocrine organ. It's secreting compounds that dictate your body composition, your energy balance, and ultimately your entire trajectory for healthy aging and longevity.
Let's jump to Section 4, Pathophysiology of Irritable Bowel.
So the Rome 4 criteria builds our clinical framework here. It defines IBS not just by altered bowel habits, but by recurrent abdominal pain tied specifically to defecation. And we see different subtypes based on motility, right? IBS-C for predominant constipation, IBS-D for diarrhea, and IBS-M for mixed. But look, as systems thinkers, we can never, ever treat this clinical diagnosis as a hard endpoint. We always have to dig deeper and ask, what is actually driving the dysmotility and the pain at a cellular level?
The thing is, IBS is not merely a mechanical digestive issue, it's actually a profound gut-brain interaction disorder. Patients exhibit visceral hypersensitivity, meaning their enteric nervous system is hyperreactive. And we know this is heavily driven by an overproduction of mucosal serotonin, or 5-HT, right there in the gut. When mucosal serotonin gets dysregulated like this, it wildly alters gut motility and transmits these super exaggerated pain signals to the brain, just completely disrupting that entire gut-brain axis.
Which brings us perfectly to Section 5, Neuroinflammation and Mental Health. So, the crucial point is tracing this neuroinflammatory pathway. Let's look at Step 1. Pro-inflammatory cytokines generated in a leaky gut travel systemically and literally breach the blood-brain barrier. Step 2. Once inside, they activate microglia, which are the brain's resident immune cells, specifically within the limbic system. Step 3. This neuroinflammation alters task-based functional connectivity in the brain. And finally, Step 4. This physically increases the reactivity of your amygdala, resulting in significantly heightened clinical anxiety. So gut inflammation is directly causing psychiatric symptoms. It's this continuous, completely destructive feedback loop. But here's the good news. We can actually intervene in this loop through targeted sleep and adaptogens. Empirical clinical data shows that specific psychobiotic strains, like Bifidobacterium longum 1714, can actively modulate this axis. By altering the microbial ecology, this particular strain significantly improves sleep architecture and duration under chronic stress. And remember why that matters. Deep sleep is when glymphatic clearance occurs. That's the process where your brain literally flushes out metabolic waste. So by improving sleep quality, the microbiome is facilitating critical neurological recovery. And we see similar systemic effects with another targeted psychobiotic, Lactobacillus paracasei PS23. In a really interesting study of highly stressed clinical nurses, this strain didn't just improve their subjective mood, it actually caused a significant, measurable reduction in circulating systemic cortisol. By acting as a biological adaptogen, the targeted microbiome literally downregulates the HPA axis, interrupting that initial stress cascade we mapped out earlier right at its very source. Finally, Section 6, Diet, Psychobiotics, and Interventions. Bringing all of this systems biology into practice really comes down to dietary and microbial modulation. Interventions like the Mediterranean diet, which is incredibly rich in diverse fermentable fibers and polyphenols, act alongside these psychobiotics as master regulators. Together, they increase microbial diversity, which dramatically ramps up that SCFA production we talked about. This strengthens the intestinal barrier, lowers your systemic inflammation, preserves metabolic flexibility, and ultimately feeds this virtuous biological cycle that restores homeostasis across the entire network.
I want to leave you with this really provocative thought straight from the bleeding edge of neurogastroenterology. If tight junction proteins like Claudin-5 govern the blood-brain barrier, could future modulators allow us to intentionally open and close it? Just imagine that, temporarily opening the barrier to deliver targeted Alzheimer's drugs, or sealing it tight to prevent gut-derived neuroinflammation. The map of the microbiota gut-brain axis is expanding every single day, and we are just beginning to unlock its true therapeutic potential. Thank you so much for joining me on this deep dive. Keep questioning, keep learning, and I will see you in the next explainer.